Okay. Okay, so we are now recording, and thank you very much, Pinar, for agreeing to give a Build-A-Cell seminar, and it's all yours.
Thank you, Kate. First of all, many thanks for inviting me to give this talk in the seminar series. I'm very excited to share what we have been doing on computational work with cardiac cells with a broader audience here. And I am a professor of aerospace and mechanical engineering at University of Notre Dame. And I work mostly on bioengineering area. And the type of work we do in my lab, except this specific project that I'm going to talk with you, is on more biomedical applications of cell-cell and cell extracellular matrix interactions. We are interested in many diseases, but heart diseases in specific. And so some 4 or 5 years ago, we started to explore using heart cells as one part of the projects that we do in my lab for biocomputing applications. I have been working with heart cells for a while now, and they're fascinating. And Kate just asked, what's this picture here? So it is a zoomed up image of single heart muscle cells striated interstructure that allows it to beat and contract. So in this particular project, we were very interested in using heart cells for biocomputing applications, and the motivation for that came from the fact that heart cells are very much like neurons. They are very similar in terms of the membrane potential dynamics. They can produce action potentials, and obviously they are not as adaptive as neurons. They don't form new junctions based on learning or similar methods that the brain grows and establishes itself and changes itself. But for this particular area of biocomputing, we thought it may be an advantage, because then we have a robust building block that is excitable by electrical signals from the outside world. So it can potentially interface with traditional electronic devices. And also it is reliable in the sense that it is a steady, it has a very steady membrane potential profile as it beats, and it can potentially be used for engineering in a more reliable sense than neurons, because they wouldn't try to adapt or learn by themselves, and they would just be doing the tasks that we will assign them to. So in this particular talk, I will talk about our research in using these cells as oscillator units, so coupled oscillators that would form networks of beating muscle cell clusters. And this is a collaborative project in between my colleagues in electrical and computer engineering, Suman Datta of Notre Dame, and Nikhil Shukla, who was a former student of Suman, who worked on their collective computing algorithms, and now he's a professor in University of Virginia. So this I'm not a computer scientist. I don't develop algorithms. So we mostly worked on the hardware, trying to understand and experimentally create these networks of muscle cells. And Suman and Nikhil helped us understand and map them to these collective computing algorithm devices that we were able to produce using these beating heart muscle cells. But in a more general sense, I would like to introduce what collective computing is. And as I said, I learned this as we started to work on this project. So in current paradigm of things, the algorithms that are used with the current electronic devices and computers, they all rely on Boolean logic. And I'm sure the Build-A-Cell community is very familiar with the Boolean logic operations, because it is one of the well-established areas under synthetic biology. People have been using gene and protein circuitries for biocomputing, and that's how we first got inspired. The logic gates based algorithms, they tend to search every single solution for a problem, and that may be okay for some applications, but some applications in computing requires the global minimum or global optimal solution for a specific problem. And the Boolean-based algorithms tend to search the entire solution space. And when you have a lot of nodes in a network, for example, where you try to find a certain, and I'll give an example for that. One of the most common used examples for these type of problems is graph coloring. And I'm gonna kind of build the case around that one, but the solutions that we were able to get can be applied to other problems of the same sort. So this kind of problems are called combinatorial optimization problems, where your system can have other optimal points, but the global optima can be found by the Boolean algorithms after searching the entire solution space. It takes a very long time, but nature does this spontaneously, and the oscillators and coupled oscillators are one example for that. And for this very reason, collective computing using coupled oscillators have been explored in the computer engineering community, as well as in labs that use traditional silicon-based devices. So there has been attempts to use qubits as coupled oscillators. There have been optically coupled oscillators, phase transition oscillators, which are made from vanadium dioxide, as I show here. So this is a work by our collaborators Suman and Nikhil. So there's silicon-based devices that this collective computing has been explored already. There's spintronics and there's the CMOS oscillators, obviously. So and how it works is as the oscillators couple and converge to a stable frequency, the phase information in the optimal state of the coupled oscillators, which is when they are all coupled together, corresponds to the optimal solution of the physical problem that we are trying to solve. And in our case, we wanted to repeat that with bio-oscillators and treat the cardiac cell as a beating oscillator. But I want to give some background on these cells, because I know some of you probably are familiar, but maybe some of you don't know the basics of heart cell phenotype or behavior. So when we first started thinking about muscle cells as computing elements, the reason why we got inspired by this idea was because heart is composed of mainly two cell types, the fibroblasts, and here shown in red, and the cardiomyocytes, which are the muscle cells shown in green here. And although throughout the heart they are coupled together using these through these surface proteins channels, connexins, so ions can pass in between them. However, only the cardiac cells can respond to outside stimulus. So this simulation very nicely shows how, if my video works, oh, cannot play the media. Okay, so I'll just explain because the video is not working here. But so when a signal hits a patterned cardiomyocyte fibroblast structure, it could be as simple as this little rectangular pattern here. The heart muscle cell side, the cardiac side, receives the signal and in response creates an action potential that propagates throughout the cardiac side. And then once it hits the boundary with fibroblasts, it starts attenuating, because at that point, still the ions can pass through internally in between the cardiac cell and the fibroblasts, and to a certain degree within the fibroblasts too, but it cannot be regenerated. So a new action potential cannot be produced. And this was the first idea around this, using heart cells as computing elements. And this was a diode analog that we created using the heart muscle cells, where the signal that comes from the cardiac side can pass through the diode, because it will still be detectable at the end. If the simulation worked, I could have shown you. So there's the boundary, and once it hits the boundary, the signal attenuates. But depending on where you measure it, you can still have a output signal in the cardiac fibroblast side, and the same degree of signal coming from the fibroblast, and doesn't have an output on the cardiac muscle side, because fibroblasts cannot create the action potential in response to that stimulus. And this video shows the diode where the half of the pattern beats and regenerates the action potential, or in response to an electrical signal that we provide using these microelectrode arrays, can propagate the signal till the other end of the device, while the same kind of signal cannot propagate back because the fibroblast cannot create the action potential. So this was like initial proof of concept idea of using heart cells as computing elements, and then we went ahead and did some AND gates and OR gates, all inspired by what your community has been doing in terms of using synthetic biology and synthetic circuits for by computing. But while discussing this with colleagues in electrical engineering and computer engineering, they suggested that this is already, I mean, the biological entities are already on the slower scale. And if you want to use them for computing that can potentially compete with current technologies, then using something that is inherently good about them would be a better approach, which really made sense to me at that point. So we decided to switch all the gears from Boolean logic-based devices and trying to use the heart cells, which is possible to create all those kind of circuitries. We decided to use them for what they do best, which is beating at a steady phase and then coupling in a locked steady frequency, which can be mapped using the similar algorithms that the collective computing community has been using with silicon-based devices. And I'm going to talk a bit more about what's the graph coloring as the toy problem we picked for this exploration of using cardiac cells for biocomputing. And that's the task of assigning colors to a graph, to a map of, for example, United States. And the constraint is you want to use the minimum number of colors that would color the map, with no neighbors having the same color. So that's a example constraint optimization problem, and it is easy to see for a few nodes of the graph, and like 3 or 4 nodes. But as the graph gets more complex, this becomes a NP hard problem, and beyond certain nodes, it is very hard for the current algorithms to solve. It takes days and days, and this has very meaningful real-world applications where you can use it for resource allocation or pattern recognition even. So there is a wide range of different types of problems that supply chain, artificial intelligence, transportation, and bioinformatics, computer vision, you name it. So all these problems fall into this category of combinatorial optimization problems. And in this particular project, we have been using this graph coloring as one of the toy problems that falls under this area of combinatorial optimization problems. And there's a huge deal of mathematics going behind it, and there has been like groups that specifically, as I mentioned, try to use different types of oscillators for this purpose. And so far, the CMOS ones looks more promising. And in either platforms or materials that people use, the idea is all the same in terms of the math that goes behind it, which I'm not going to go through today. But as the oscillators couple and lock into a steady frequency, the order of spikes of each beat or each oscillation can be mapped to the optimal solution. So, I will show you how we fabricated these devices using heart muscle cells as a potential new platform or media for to be used in collective computing applications, in addition to the current ones that have been already explored. So in our case, our elements are rat cardiac muscle cells or rat cardiac fibroblasts. So we have two elements. And as I briefly introduced in the diode idea, we had the muscle cells are electrically active and they can regenerate the action potential that keeps them beating. So they can beat forever as long as you give them some sugar. And that will be probably energy is one of the main highlights of why this system can be a good alternative to the current CMOS or other types of oscillators. So they are really energy efficient. And then the cardiac fibroblasts that are the passive connectors between the beating cardiomyocytes, they are also essential. They are essential to create an RC coupling in between two beating clusters. So the problem can be mapped to the network that we are trying to analyze. So in experimental setup, and I'm going to show you some of the experiments we have done, the graph coloring problem is mapped to a device where we can pattern and control the location and connectivity of different clusters of cardiac myocytes or oscillators in this case, we call them. So this is an example for a 4-node device where you have all-in-all connectivity. If you have all-in-all connectivity, then the answer is 4 different colors because no neighbors can be the same color. But physically, what we are doing is we are creating patterns of proteins that would constrain the cells into those locations. And we are blocking first the bridges that would connect them specifically using micro patterning approaches. Then the nodes or the bioscilators are seeded with a mixture of muscle cells and the fibroblasts. So as in the heart, a presence of few fibroblasts in these nodes would not affect the oscillator behavior of the cardiac cells. In fact, it makes them stronger. So they are beating stably with the combination of like 30% of fibroblasts and the rest cardiomyocytes all confined to these nodes. And during the initial seeding, the bridges are blocked. And after the oscillators start beating, we remove the bridges, we remove the blockers at the bridges. And the fibroblasts in between the oscillators start to grow and connect these individual oscillators. And then the idea there is once they start all beating at the same frequency, after they will couple, the phase order in the beating profile of these different oscillators would be able to, we would be able to map it to the number of colors that we need to assign to this kind of graph. So that was the initial idea. And for that, we have done a lot of modeling and experiments and simulations. And the modeling part is mostly done with Jorge Gomez, the student of Suman Data, who is about to graduate these days. And he used SPICE modeling and modeled the biooscillators, the muscle cells, as relaxation oscillators that is coupled by a fibroblast bridge that has RC type of coupling. That's like the requirements for this whole thing to work as a relaxation oscillator and able to do computing with the coupling dynamics that we would extract from that. But once when we started this project, we didn't know, and there was no literature on it, if the cardiomyocytes would act as a relaxation oscillator or not. In other words, can they independently beat at two different frequencies, and then upon being connected by a fibroblast, would they converge to an independent and steady frequency? So instead of having a master-slave kind of relation, can they act as truly independent oscillators? So that's the first thing we tried to figure out. And for that, two of my students, actually graduate students, Yang and a postdoc in my lab, Shang, have worked really hard to do these very tedious experiments with cardiomyocytes, and we started very simple. We use these microelectrode arrays that has a blocker placed. I mean, we created these blockers with defined widths that we separate two clusters of heart cells, and then we assigned them, we treated them as oscillator 1 and oscillator 2. And after oscillator 1 and oscillator 2 starts beating, we remove the blocker and we let the fibroblasts grow and fill in the gap and connect the two oscillators. So the cardiac cells are non-proliferative and mostly non-migratory. So they stay put in the initial areas we assigned for them, and then the fibroblasts, being proliferative and migratory, they fill in the gap and form the bridge. And this is important because if we end up having cardiomyocytes in the bridge, then the two oscillator becomes one, because there won't be any lag in terms of the phase that these two oscillators are beating at. So they will all act as a single one big oscillator, which we don't want, because we want to be able to see the different phases after they all end up in the same frequency. And that is our solution for the graph coloring. So this is the experimental results for different width separations of two beating clusters that are, in this case, very primitively fabricated, just to see what would be the correct width of fibroblast insert that would give us enough resolution in terms of the phase differences that we are getting from these two beating clusters of cardiomyocytes as they start synchronize. And we have done very long-term recordings up to 2 days. And first of all, the good thing to see was the beating was, for the most part, very steady. So these are actually reliable oscillators. We have seen width-dependent phase landscape in these different width inserts of fibroblasts that we tried. Also, this is kind of characteristic of a relaxation oscillator that can be made using silicon technologies. So that was also very reassuring to see, in the sense that we have a phase and frequency landscape here that we can work with. And it is possible because the fibroblasts actually act as a RC type of connector. In this experiment, we also measured their impedance, and we saw that they have a nice impedance profile that shows us that they're truly RC type couplers, and they can be used as the coupling elements for these two clusters of beating oscillators. And the difference in between, the phase lag in between the two clusters, can be adjusted by the fibroblast insert. So that's a trade-off in between the speed of computation and the resolution of the output. So if we use a too short of a fibroblast length, the coupling happens much faster, but it is harder to distinguish different phases. And as the node sizes increase, this will be a kind of a trade-off that we will have to decide. We did decide to stick with 300 micrometer for now, but as we grow for more than 64 nodes, which is where we are at, that I'm going to show you some results of, this may need to be recalibrated. And after establishing the basic properties of these new computing elements in terms of their impedance and the pairwise coupling dynamics, we went ahead and started to use more elaborate fabrication methods to create more defined node sizes that are accompanied by mobile blockers that can be more precisely placed, and also electrodes that goes directly to the bridges and the nodes as we want them to be. So this was all fabricated by Jiaying, the brilliant graduate student who was working on the project. And we were very happy to see that we can actually solve a 3-node problem. This was one of the prototype devices, the first demonstration of you can use the hard cells for solving a graph coloring problem for 3 nodes. But I'm saying that because after they locked to a steady frequency, and each color here, green, blue, and red, shows and this is the Fourier transform of the whole recording, they all locked to the same frequency around 2.1 hertz. But if you look at the waveforms, you see that there is 3 separate phases, and the phases are separated with enough distinction thanks to the 300 micron insert of fibroblasts, and the optimal solution to 3-node is 3 colors, because they're all in all connected. And obviously for 3 nodes, it is easy to see, and it is not very hard for the Boolean algorithms to compute. But we also ran some simulations and saw that the scaling of the cardiac oscillators is much better compared to the current gold standard Boolean algorithm, the Johnson algorithm. And for the cardiac oscillators to beat the Johnson algorithm, we need to really work beyond 64 nodes. So you really start seeing the differences when you have 126 nodes and beyond. And this cardiac cells scale really slowly, while the Boolean algorithms really go exponentially to MP hard as you increase the node sizes. So the next challenge was to create much larger networks. But since we showed that with the 3-node it works, we were very hopeful that maybe we can create larger networks and still get the optimal solution. So we kind of scaled up our fabrication, and this shows a 9-node fabrication. The scheme is the same. We pattern the proteins, and then we place the blockers to block the bridges, and the cardiac cells see that the nodes start beating, as you can see in this video here, after a while. And then after they start beating, we remove the blockers, and the fibroblasts fill in the gaps. So you end up having this connectivity, and then this immunostaining nicely shows that the troponin, which is the indicator of the muscle cells, is localized to the clusters. So there's no cardiomyocyte cells in the bridges, while the vimentin, which is the indicator of the fibroblasts, is kind of all over the graph as we expected. And then we have used a similar methodology and expanded to 21 and 64 nodes. However, as we increase the node size, it is not feasible to create microelectrode arrays to keep using the microelectrode arrays for detecting the beating profile of these cells. And it kind of defies the purpose, right? We are trying to go away from the silicon technology. We are trying to provide an alternative media or platform for doing all these computing. And for that, we resorted to imaging instead of recording directly from the membrane potential recordings. And we have created a wave extraction algorithm that would create the beating cell videos and map it into a fitting waveform and extract the phases based on that video image analysis and fitting. And we also compared side by side this with the optical video phase extraction method we used with the microelectrode arrays that we optimized the system in the past. And we have seen that there's just a 5 degree of error in between the two. And we concluded that optical video phase extraction that we optimized can be used for analyzing the large scale networks coupling dynamics, because 5 degree error is within reason, given that we need at least 20 degrees of phase difference in order to separate and define different phase clusters in our networks. And we were able to use the imaging and imaging method for analyzing the network dynamics. And we solved the 9-node problem after the 3 nodes was successfully solved. So three color is the optimal solution for a 9-node network with this kind of connectivity. And our phase ordering showed this, the graph is solved optimally. And obviously we are trying to do 64 nodes is the largest that we fabricated. Just to put in things in perspective, some of the examples I gave in the earlier introduction, some of the types of other types of oscillators are in the order of 4 to 6 oscillators. Some of them are in the order of like 500 to 1,000. So it is not trivial to create these connected oscillators, even with silicon-based technologies. So 64 node is a reasonable size. I mean, it can be bigger, obviously, but a reasonable size of network, even within the realm of silicon-based technologies. That's what at least Nikhil Shukla told me, who works on the silicon-based side of things. So we are proud of this 64 node network that we are analyzing through the sub graphs of it. And the reason for that is we can image currently 12 nodes at once. So we are imaging and sliding and imaging and sliding. So it is like a patchwork of data right now that we are trying to put it in the same time scale. And for that, we are optimizing and developing other types of data analysis methodologies. We have overlapping nodes that we use as a frame of reference. As we image and slide and image and slide, we make sure that there's a degree of overlap so that we can trace the frequency and the phase information. But the subnodes that we have been analyzing, and this is, I think, the result of the 12 node, but up to 32 nodes right now, we were able to put that patchwork together and can get the optimal solution. So where are we going with this and how does this compare? So first of all, because the cardiac cells are not as sturdy as the silicon or CMOS ones, there's a degree of error. And so this graph here compares the CMOS oscillators, the Johnson algorithm, the optimal solution. And as you can see, our cardiac cell oscillator is slightly drifting from that. However, Nikhil Shukla has developed a post-processing algorithm, which is very simple. Essentially, what's happening is our cardiac-based network ends up being stuck in a local optima, very near to global optima. But because we can go back and check this post-processing algorithm with that correction, which is a simple and easy thing to do, brings back the cardiac oscillator network to the level of the Johnson algorithm and the CMOS accuracy, while having the scaling is, as I mentioned, is much better, similar to the other types of oscillators. So this graph compares the all of the current possible algorithms and different devices, but here also we compared the CMOS oscillators and the cardiac oscillators. So the one advantage that the cardiac-based oscillators, the bi-oscillators would have is in terms of energy. So the energy requirements is much smaller compared to the CMOS-based ones, while CMOS-based ones are much smaller, obviously, and the integration may be more easier. However, I believe that the cardiac-based ones would have other advantages like self-renewal, self-healing, and also integrating them with other biological entities for potential interactions with living tissues or molecules. So with that, I would like to thank to my research group, my collaborators, and then my funding. And thank you all for listening, and I'd be happy to answer questions that you may have.
Thank you so much. I have some questions, but you have some questions in chat. Can you see chat?
Sure. Should I just read an answer or?
Yes, if you could read the question, that would be
Okay.
great. Thank you.
Okay, so thank you, Harold, for finding it fascinating. But the question is, could you do a network like that with other cells with any less consistent electrical activity? So it is possible to do network like that with other cells, but I think what's unique to heart muscle cells and the fibroblasts is that they very nicely mimic the resistive and capacitive properties of silicon-based systems. So you kind of need both, and these cells are inherently can be connected with each other and still retain their resistive, capacitive, or both type of properties. But I would love to hear from any one of you that may be interested in adding or manipulating the channels on these cells or potentially stem cells. I'm actually looking for synthetic biologist collaborators to really like fine tune those resistive and capacitive properties, which would mean that you need to decide on what kind of channels, and then once you decide, you need to make those cells produce those channels. So the answer is yes, probably can be done with other types of cells, but I don't have the tool set to convert those cells to the correct resistive and capacitive properties. If anyone is interested, please email me. Another question, what is the lifespan of the cells in your study? So the rat cardiomyocytes, oftentimes, because they end up getting overpopulated with the fibroblasts, they are stable and steady for a week or two. But we also use induced pluripotent stem cell-derived cardiomyocytes in the lab. The stem cell-derived cardiomyocytes, the longest I have in the lab has been going for 14 months. We haven't gone beyond 14 months, but they can go on for months if you use the right type of cardiac cell. Do those cells divide is another question. The fibroblasts does, the cardiomyocytes don't. So that's actually one of the reasons why the rat cardiomyocytes can't go on for a very long time, because the fibroblasts dividing kind of overpopulate and make them their beating properties not so great. Does the synchronization change if you take 2D monolayers to 3D? Yes, so that's the next thing we want to do. We want to really take this to 3D, partially because that's one big advantage that the cells would have over CMOS, because they would be able to, if we give them the framework to crawl in and populate, they would be able to form 3D connections much more easier than 3D fabrication of silicon-based oscillators. And why Connexin 43, in terms of visualizing, they have other connexins too, but Connexin 43 is one that both fibroblasts have in between them, and cardiomyocytes have in between them, and cardiac myocytes and fibroblasts have. So that's kind of, we use that for visualizing all of the possible connections, but there are other gap junctions that these cells utilize. Another question, could you connect heart cells with other kind of cells with channels or via small molecule chemicals? Yes, it would be possible, I think. And if anyone has any ideas, I would be more than happy to talk and explore. Can spiking neurons be used similarly, or use neurons to control cardiomyocytes? Spiking neurons cannot be used similarly, because they are not, they just spike at more or less randomly compared to cardiomyocytes. Cardiomyocytes have a stable frequency, so you need to, the prerequisite for using as oscillators is you need to have those oscillations happen pretty much like steadily, and neurons can create similar spikes, but they are more sporadic, and depending on, it depends on the confluency, it depends on many other factors. That's why we decided to go more with cardiomyocytes in this, but they can potentially be used to control the cardiomyocytes, yes. Can you use not just single cell cultures, but an organoid? That's a possibility, but the idea there for using 2D cultures was you want to keep this as small as possible to be able to compete with the size requirements for silicon-based technologies. So there are the oscillators in CMOS, they are like 65 nanometer, the current state of the art. Our cell is already 20 microns. So we even tried with less number of cells, but the problem is if you have too few cells in the cluster, they are not stable anymore. The beating becomes more random, because they cannot sustain a colony of beating cardiomyocytes. And organoids would give more steady beating, but you would give away from the, both in terms of the size, you would need larger structures, and also recording may be harder, because then you will need to record from different parts of the organoid and decide which part is the representative of the whole structure. Can cells communicate signals with silica parts? Yes. I mean, potentially these cells can be interfaced with the current type of electronics, and that's what the microelectrodes are doing. Actually, we can record and stimulate them from those microelectrode arrays, and interfacing is one of the advantages, I think. And how, can you please clarify how you choose the signal for each node? Does the type of signal change the synchronization? The signal we choose is based on, so once they are synchronized, the cluster all beats at the same frequency and same, how can I say? So the signal is just coming from the beating frequency of the whole cluster. So we don't really choose, but we analyze it with the microelectrode arrays. It is the change in the membrane potential, and with the video recordings, it's change in the intensity of the beating cell videos. And it doesn't change the synchronization, because synchronization happens in between the clusters, and the direct connection between fibroblasts enables the synchronization. The recording doesn't interfere with their synchronization. We are So what sort of modeling are you seeking now? So we are looking for expanding this to 3D right now, and also one other big direction we are going is reconfigurability. So we are trying to come up with systems that would allow us to reconfigurably change the network connections. So not only how one type of graph hard-coded, but also be able to break the bridges and reform them is what we are hoping to do next. I think I exhausted all the questions. Thank you all for also saying all the comments about the talk. I didn't read them, but
I have a question which might be kind of outside of what you're working on, but there's a lot of efforts in our community on building synthetic cells that resemble natural cells in function. Would you be able to speculate how many genes would we need to make a synthetic cell that displays some sort of a coordinated electrical activity?
Oh, that's a hard one. I can't totally speculate, but on top of my head, maybe 30, 40, up to maybe like slightly more than that, but I would guess at least, because there are like ion channels and like all the intercellular calcium handling and everything. So yeah, at least 20, 30. Is it doable? I don't know. It's too much, right?
It's a lot, but it's not crazy. I honestly thought it would be much more. I was expecting an answer in hundreds.
I mean, it is just like me thinking all the chances I know, you know, all the like ballparking. It could be up to 100 easily, but I wouldn't think that it would be 100, you know?
Yeah, that's really fascinating. So thank you so much. Thanks everyone for the questions. Thank you, Pinar, for answering all the questions. And
thanks for inviting, I mean, and giving the chance to me to share this with a broader audience. And I will try to attend the future talks. You have been doing this seminar series with amazing people. Very happy to be part of it.
Thanks. Bye, everyone.
Bye.